Hey guys, I'm Albert from ValleyDAO, and as you can see here, we're in London. During this time, we'll be meeting up with our funded researchers at the Cocoa Zero project and check in on how the research is going there. Yeah, so what is happening here with these shaking flasks for...
So this is our incubators. They're literally just shaking in there and being heated up to a nice cozy 30 degrees so that the cells are happy and growing and well mixed, so they get enough air in there and enough nutrients are mixed, and yeah, they grow in here for 2 days, 3 days, 4 days, as long as you like it. And then you take the shake flasks out, take your samples, and continue with our review.
In the context of the Cocoa Zero project, what do our cells usually do in these?
Our cells really enjoy the shake flasks.
They do.
Because if you are in a well place, you often get aeration problems. So they don't mix as well. They don't get as much oxygen. The Yarrowia is strictly aerobic, so they need oxygen. So having them in well plates is usually quickly limiting for them because they do grow to higher densities and don't need their oxygen to live and to produce lipids.
Yes.
So we are usually in 100 milliliter shake flasks. Is where I screen most of my experiments, and when it is upscaling, then a bit bigger shake flask. Here is frozen biomass that we have cultivated in the bioreactor and then harvested. And then we have put them through a dehomogenizer. So we have broken the cells to release the lipids, and then we can extract the lipids using different solvents and then measure the lipid content, measure the lipid composition, or use the lipids for whatever we like. Cocoa Zero.
Cocoa Zero.
So this is our culture that was very clear yesterday, and now you can see all the cells in there. So this cloudiness is all little Yarrowia cells, yeast cells that are growing in this media and that are apparently very happy. So this is just 1 day old. So I'm going to give this another 1 or 2 days, and then I can continue with that.
Yeah, and this is just a very small culture
Yes.
compared to what has been like scaled to currently.
Exactly, we have scaled this.
Fortunately, we don't have footage of that, but
no, we have cultured this in a 30-liter bioreactor with a volume of 12 liters in the end. So this is just 10 milliliters. So this is significantly bigger.
Yeah.
Yes, we have our fume hoods here and the chemical hoods. So that anything, if you work with solvents or anything dangerous, you put it under here. You work with this constantly ventilated, so you're not releasing it into the environment, aka our lab and us. So we're not being exposed to it, and that's being filtered and taken care of.
Yep.
So this is where we do the lipid extract, the chemical work. Basically.
Staying safe.
Exactly.
Yep.
So this is the GCMS.
AKA your second wife.
Exactly. I guess primary wife, because I don't have any others.
So for context, a big part of the project is like analyzing the fatty acid composition. And so Oliver is spending a lot of time here. And if you got the vials here. So each vial contains a sample, and I guess it's injected through the tubing system in the machine, and then it gets separated.
Exactly.
And then analyzed.
I can show you actually a bit more of it.
Okay.
So we can have a look into the machine if you like. Because it's actually quite a nice machine. So how this thing works is that, as you said, the sample is being injected on the top. It goes through the system. There's a guard chip that protects the main column from dirt. It goes through here, and this little thing, this little thread of copper basically is the column. So this is 30 meters of column in which the injection will interact with a solid phase.
Yeah.
And depending on the temperature, this is a little oven, a very efficient little oven.
Yeah.
It goes from just 30 degrees to 300 degrees and can also change temperatures super quickly. And depending on temperature, your components will interact with the solid phase more or less. They will be released and then separated based on what they are. So you can separate a mix of different compounds to single compounds that come out.
Yeah.
And then go over here into the next machine, which is the mass spectroscopy. Which basically looks like this. So it's a very complicated little machine. It's all in vacuum. It's always using helium, very high quality helium as a carrier gas to bring this in because it's not reacting with anything. And yeah, all of this will then result in some nice peaks on the computer, and then will tell us how much we have of which compound.
All right. So yeah, as we mentioned before, the GCMS is a pretty big component of the project because we're constantly cultivating new variants of the yeast cells, and then they go on and then they produce the lipids, and then those have to be analyzed. And yeah, the ultimate goal is to get the fat composition to mimic that of like from natural occurring cocoa fats, like from the plant. In this way, we don't have to ship these fats halfway across the world. We can hopefully produce them locally, such as in London or maybe outside of London in the UK, and then we can make chocolate or use them for cosmetics, for example. So yeah, I mean, there's so many. I mean, fats is essential component of like everything. It's used in so many aspects of our everyday lives.
So this is the freeze dry. So in here's vacuum, and down here, that's where there's something condensating, is a cooling column.
They look like sheikhs, you know, Middle Eastern sheikhs with their little bandana on them.
Yes. Lipid samples go through here because for the conversion of fatty acid to fatty acid methyl esters, you cannot have any water around in the samples. And of course, your cells are full of water. They were grown in media, which is mostly water. So you have to get rid of these things and you remove all the water from it. And this is happening here. So you have vacuum and you have a column that is minus 60 degrees. So all the water.
But this is minus 60.
Not here, but it's connected through here and down here. If you touch here, you feel that it's very cold. That's why it's also all the water condensing. Down there is where the water is then condensing again.
Wow.
And this is how you get rid of all the water. So that's where these samples look like they are kind of dried out and shrinking over here. So because people left them in there for probably a day or two or something, and then remove all the water from it, and then can continue with the
freezer.
Exactly. So this is minus 80 freezer. So this is where we store our cultures in different strains in the long term. And then they can stay for years and years, and they're in glycerol, so they are not completely dying. They have a good chance to survive, but some of them. And then you can take them out, streak them again on a plate, put them in the liquid media, and they do actually recover and grow again. And this way we can test strains even after years, because usually they would grow on your plate for a few days, and then they will start dying, so you cannot keep a fresh sample for years.
Whoa.
A lot of ice.
Whoa.
A lot of samples. So each of these little spots is essentially a rack in which then there is more and more samples. And this is what this looks like.
Boxes within boxes.
Boxes within boxes. Shelves and shelves, and with a lot of little vials. So this is literally thousands of different samples we have. We have, of course, an Excel sheet that organizes this, that you can find your sample, because if you don't know where your sample is, we're not finding it in here. So we know which sample should be in which box and where the box is, and then you can find your sample. In here you also have the rest of our collection in here, which then you can take the boxes out.
Right. So just a different layout.
Exactly.
Yeah. We don't need to do it.
Yeah, everything is for the pros.
Yeah.
Use this. Also doesn't seem like the way it should be functioning. Like cultures here.
Yeah. How many researchers do you have in here currently?
So I think our lab, so this is a shared space. This is not just our lab, this is the Center of Synthetic Biology, or now it's called the Center of Engineering Biology.
Yeah.
This is like 5-6 different PIs.
Okay. In just this...
In this shared space. And we altogether like 50-60 people or something.
Yeah.
The RLA lab, so the Rigorous lab, is 30 researchers-ish. Changes. There's visitors coming, there's people finishing, people starting.
And there's another section similar to this, like a copy of this.
On the other side, this is a teaching lab, actually.
Okay, students.
A copy of this one, also very big space, very empty, because it's only used occasionally for teaching. It's very sad, because we are cramped in here, like we want to have some more space.
Yeah.
But no, this is for teaching purposes only, and that's why they are not allowing us in there.
Yeah.
But yeah, this is our lab, our shared space.
Yeah.